I think Roy was up first, so I'll defer to him.
Yes, Roy Herbst from Yale. I have a question for Spyros. I like the idea that you're recording the evidence and building the model. The question I have is you talked about whole genome profiling, which is, of course, not always possible. There are many other approaches being done right now. Realizing we're still in a discovery mode, would you recommend that be the technique that's used, or could there be more focused techniques or panels done at different centers?
So it really depends on what the question is that you're trying to address clinically. We chose whole genome sequencing for the triple-negative breast cancer trial and for the melanoma because when you do panels, you miss a lot, right? And a lot of the changes that I showed you that led to miraculous drug responses, we would have not picked up with any of the available commercial panels today. So one issue is that it depends on how comprehensive you are. Now, if you're stratifying patients into a clinical trial and you have a limited amount of information about the drug mechanism or a set of drugs and you know the mechanisms, then that's a terrific way of constraining the problem and stratifying patients on using that information. But when you start to think about the bigger vision, if you start to think about thousands of cancer treatments and not knowing all the information, where it's feasible, whole genome approaches to look at all of them, all of the possible changes is the way to go. The other thing is whole genome gives you the opportunity to be a little bit more multidimensional in the type of information. So with panel sequencing exomes, you're going to miss copy number changes. You're going to miss amplifications and deletions. You're also going to miss translocations. And so what we're seeing in most cancer types, those aberrations are very important. So where feasible, we would advocate for whole genome approaches. Yeah.
Thanks.
Most of the panel has Lowell Schnepper, Boston, Beth Israel Deaconess Medical Center. Most of the panel has emphasized astonishing advances in basic cancer biology and systems biology, all of which or much of which is focused in the translational context on treating patients with the disease. That's of course understandable from the clinical perspective, which is where I come from. But Denise's point about virus-induced cancers and the ability to identify and the etiology of so many, 20% of the world's cancers, and develop vaccine approaches to preventing them, leaves me wondering whether our policies in terms of science policy for the nation should in fact attempt to redirect our fundamental the translation of our fundamental biologic observations, biomarker work, systems biology work towards prevention in a far more ambitious way than has been done to date. As a cancer clinician, I can tell you that I have very great skepticism, except for the highly occasional cancer that is fortuitously sort of identified with an appropriate therapeutic combination or single agent, that advanced cancer is going to be easily treatable as much as it's likely to be preventable if we can figure out what the right targets are.
Denise, do you want to comment?
Well, I mean, certainly cancers are the easiest target because you know the precipitating agent, you know how to look for it, you know what the pre-neoplastic lesions may be in many cases, and so you know, it's the opportune cancer to use prevention as a mechanism to get at. But even there, I would caution that something like Merkel cell polyomavirus is something that 65% of the population acquires in childhood. There are 2,000 cancers in old age. We're not going to vaccinate for Merkel cell cancers, even though theoretically maybe it would work. But we could still use that information for treatment. Now, for cancers that we don't know the etiology, for breast and prostate and colon, we've tried various prevention trials that haven't been successful, largely, I think, because we haven't fully understood the biology that goes behind what it is that goes wrong that we need to prevent. And, whether or not these single agent retinoic acid, whatever it is, will be efficacious. And so I advocate prevention because it's certainly much better to prevent the cancer than to ever get it. And I think we need a lot of research. I advocate research in that area. But I don't think it's always going to be as easy as treating or detecting or preventing a viral-associated cancer.
Bill?
So I've felt for some time that we should turn a lot of this sequencing power to pre-neoplastic lesions for several reasons, one of which is that there's far less genomic noise. Secondly, when studied, in almost every case when there's been a high-value target, such as BRAF mutations in melanoma, that mutation was present in the pre-neoplastic correlate. And so I think we want to find the earliest sort of founder truncal mutations, not only for therapeutic purposes, but to your point, at least as a thought experiment, if we had enough deep sequencing of pre-neoplastic lesions, I think it's quite likely you would find some mutations that would represent sort of an evolutionary dead end for that lesion. That would then be a lesion that had no capability of going on to becoming a malignant tumor. I think that would have tremendous impact, obviously.
Treat the cancer before it gets cancer. John?
John Mendelson from MD Anderson. Dr. Mousses' second point, change policies that limit how genomic data are interpreted and used in the clinic. I'd like to ask him and Dr. Califano to both elaborate on that, especially what policy changes are you thinking about that would allow us to use this systems approach that we can't use now because policies are wrong?
So let's begin with how we've done things in the past and where we can improve. When you look at the way clinical trials are regulated by the FDA, the types of interpretations and devices that one could use, there are very strict requirements for reproducibility. So if you create a software system to interpret how a set of features could link to the indication of a particular treatment or stratification into a particular arm of a trial, those need to be locked down, fixed, so you can study them and move them, so, and show reproducibility, and there's a lot of strict requirements in that regard. Well, the reality is that those predictors are not very good, and they need to improve. And the only way to improve is iterative rapid learning. Iterative rapid learning means that if we take a patient with a particular context, we make a prediction. If that prediction fails, we should not make the same prediction for the second patient. And we should not have to wait to treat hundreds of patients before we open up a new trial to change that particular interpretation. We should be able to integrate learning from each N of one experience to improve. So the problem, of course, that various agencies are going to see is that, well, your first patient is going to be treated very differently in terms of interpretation than the second patient, the third patient. Well, that's exactly what's going to happen, and there's nothing wrong with that. I think we need to think about this more like drug developers. We have to start to think about how do we put a patient onto a trial, what criteria do we select, and those criteria shouldn't be detailed. They should be general. They should be, if a pathway is indicated by any of these sophisticated means, then we could put the patient. But the idea of having a rules-based approach that is fixed and locked, I think, is short-sighted. And again, it's because we've been, for many, many years, working in the premise of epidemiological evidence. We want to move towards mechanistic evidence. And then the second point is experimental therapeutics are key. So most of what we're going to be doing is we're not going to be indicating existing drugs. In some cases, we'll be repositioning approved drugs. But it would be fantastic if we had an ability to approach this as a clinical drug development trial where we can get access to drugs. And I think more relaxed rules about using experimental therapeutics and better interpretations combined is going to be useful. Andrea?
So let me add briefly to that. I mean, one of the fundamental problems that we have right now is that it's very difficult to get IRBs to approve the concept of a trial where the actual specific pharmacological agent or agents are not defined a priori. And sort of one possible way to go around this is to use bucket studies. The reality is that in many cases you will not know what those pharmaceutical treatments are until the patient has been analyzed. So it's a kind of a catch-22. So I think that there has to be some fundamental rethinking from the perspective of institutional review boards of how this type of approaches can actually be spearheaded and brought forward. The second fundamental problem that we see is access to therapeutics. So for the one that are FDA approved, it's relatively straightforward, though not necessarily always reimbursable by insurance companies. But for instance, late-stage experimental compounds, or even early stages that have been, for instance, tested in clinical 1 and for which much data is available, it's extraordinarily difficult to get access to those therapeutics through pharmaceutical companies and biotech. And again, for instance, what CTAP is doing, I think, is breaking new ground in potential increasing the availability of those pharmaceutical compounds. I think the fact that right now patients have to go through this harrowing experience of having to request sort of compounds by writing letters to pharmaceutical companies for compassionate use is absolutely sort of ethically and also from a point of view scalability untenable. I also want to make sure that one thing becomes clear, because this is always confused or misunderstood. When we say N of 1, I don't think any of us envisage treating cancer in the future on an N of 1 basis. I mean, it would be crazy to think that we will actually have to treat patients one at a time. It would not be scalable to any significant amount. The reason we're doing N of 1 today is because we need to understand this problem of tumor heterogeneity at a much deeper level. What we tend to find is that about half, more than half, than the drug or drug combination that we predict using our systems approaches, and not just ours, but in some cases those of our colleagues, are in fact very successful in a preclinical model. So you can predict from the about 25,000 square possible combination of genes, or 1,500 square possible combination drug, just the top 10, and 5 of those top 10 will actually work. Okay, which for me is just, even 5 years ago, I had no idea this could even be accomplishable. The problem is that you still don't know which one of the 5 that work in a patient, and which one of them that work repeatably when you actually go across a large number of patients. And so I think the reason for having these studies today is because they will inform how we will be able to stratify, and in many cases predict, as was asked by the previous person that asked a question, how we can actually predict how cancer before it even occurs.
Next, go ahead.
So Warren Kibbe at the NCI. So I've heard a couple themes run through a number of talks, and one of them was public-private partnerships. Another was moving from academia out into industry. I'm curious from an IP perspective, do we have the policies in place to let that happen, both from the federal standpoint and then also from an academic to private partnership standpoint? So I'd love to hear your thoughts. I know Bill and Ellen in particular, you had slides about that.
Well, I can just comment, and I think it's something that we spend a lot of time thinking about, and often we work with the tech transfer offices at the academic centers to see actually what's been covered, what the patent is there actually freedom to operate, or are they infringing? So we look at it not at the very early stage, but as it's entering into what we might call a development pathway. That's when we spend more time looking at it. And so we have an IP transactional lawyer who we have on staff, and we actually do spend quite a bit of time looking at those things.
So one of the things I've learned over the years is there are a lot of examples of decisions that get made in the pharmaceutical industry where it takes a tremendous amount of courage to say yes, and no courage to say no. And one example is approving an MTA. I mean, what's the upside to them? So I think there's a lot of risk when you put your neck on the block and say, I've approved this MTA for this investigator at University X to study our compound. And so the default is no, or at least it gets strung out over months and sometimes years. And this has happened to me even when I've had relationships with that company of various types. So I just know generically this is a big problem. So, we talk about it year in and year out of having some sort of boilerplate universal MTA and having some reasonable set of criteria where we agree as a society that as payback for all of the public investment up front, by the time your drug goes into, name it, phase 1, 2, 3, whatever it's going to be, you agree to play by this set of rules. And of course, err on the side of protecting the interests of the company. But I think that would take a lot of pressure off of the pharmaceutical companies. On the flip side, Mike Bishop has said, and I don't know if it's true, that on balance, tech transfer offices and universities are money losers. But I think everyone wants to have them because they all have this sort of lottery mentality that they're going to have the next Olympta or the next Enabro or whatever it is. So, they have these very expensive, cumbersome, tech transfer offices. And, they're at least half of the problem in terms of exchange of reagents and IP. And maybe we need to do a rethink on that. What's a reasonable set of rules for how we're going to exchange information, IP, and reagents?
Great comments, Bill. Rochelle?
Rizal Kurzrock, UCSD. I have a question for Spyro. I think you brought up some very interesting points. And historically, we've looked at drugs in a drug-centric way. We approve a drug, and we don't actually expect everybody to respond to that drug. And so now we have the N of 1 dilemma. And is it possible to get evidence for that by having a strategy-centered approach? In other words, could we approve the strategy of molecular matching or a systems approach? And once that strategy is approved, not every drug combination in that strategy has to be proven. So I don't know if that's articulated well enough, but I think that would help to get around the N of 1 dilemma. And I don't know if you have any comments there.
No, that's exactly right. I think that having an approval for a methodology or a systems approach to say that provided that you have the rigor of science, and it may not be epidemiological knowledge that you use, it could be mechanistic knowledge, and you put rigor around how you generate that mechanistic knowledge, then I think that is probably the best way to address that. And again, I want to echo what Andrea said about the idea of it's not always going to be N of 1. Where we want to move towards is understanding the mechanism behind the disease and aligning a drug, understanding the mechanism of action of a drug, and understanding getting the insights into how that disease is going to respond mechanistically to that drug. Once we have that methodology and it's reproducible, then what we're going to find is we're going to get away from the way we classify disease today, which is very ambiguous based on anatomical and pathological and histological classifications, more towards some, and again, we don't want to move all the way to a molecular nosology and just use molecular features to classify disease. We want a systems approach where we take the clinical and the molecular information, integrate it into a new type of classification, and once we have that type of classification, then we can go back into evidence-based medicine, but with more detailed classification of disease and better mechanisms. So I couldn't agree more with creating a strategy approach to regulating how we make interpretations.
So let me modulate for a second what you just said. I think one way to do that is also to really use in a very advantageous way some of the preclinical study that can be done rapidly in terms of sort of what we now call co-clinical studies. And so co-clinical studies, I think, that are done by just randomly trying out what potentially could work are not very informative, because something will work due to quirkiness of the specific PDX and the selection of those cells. But if you actually predict what should work from a human patient data that has never seen plastic, has never been cultivated, just directly to the biopsy, you predict it works in the actual PDX, and you're only testing, so the top, say, 5 things that come out of the study, then the probability that it will work back put back in the human context significantly increase. So I think that one way to reduce sort of the N of 1 dilemma, as you say, is to make, to change the way in which we use co-clinical studies, and certainly that's the way we're doing it at Columbia.
Eric?
So remember that I come from the NCI, which is the home of the provocative question. So that's a very provocative question. I started my training in 1973, which was at the very beginning of the war on cancer, or near the beginning. And so 40 years have gone by, and I'm a much more humble person than I was back then. And I have lived through many paradigm shifts, each of which was followed by a brief period of exuberant enthusiasm until we found out that the paradigm actually was wrong, and then we moved on to the next paradigm. So I'm always interested in hearing whether people question what could be wrong with the paradigm that they are proposing. Not feasibility problems, getting patients or convincing regulatory agencies or whatever, or getting funding, but what could be wrong with the core paradigm, if maybe one or more of you could comment on that.
Bill?
So at my most optimistic, I like to think we are perhaps where we were with AIDS when we were using single agent therapy. And again, when I'm being optimistic, I like to think that when we can put together several agents that have distinct mechanisms of action, can be given at full dose, and are not cross-resistant, we'll have the same rosy results we've had with combination therapy for AIDS or TB or those cancers where we have had a major impact. What keeps me up at night is the concern that when we've done well with targeted agents, we've been targeting more often than not the earliest founder mutation, which then was present in all of the subclones. I don't think we're going to do very well targeting later mutations that are present in a subpopulation of the tumor cells. And I think, as you know, there's increasing evidence that within the same tumor, you can find a number of different subclones that have a different collection of genetic alterations. So I like the idea of approaching what some people call the truncal mutation, but obviously there's only one truncal mutation. And when you get to the branches, I think we may have more challenges. So I hope what gets us home is combining targeted agents, and by that I mean agents aimed at specific genetic alterations, which are with therapies that are more genetically agnostic, if you will, such as the immunotherapy approaches and other things to decrease tumor burden, and to get down, to get several log kills with other methodologies, combining it with targeted agents, I think will get us home.
So the same question you asked could have been asked in about 1600 England, when people were trying to solve the longitude problem. And no, they tried a lot of different things, and every time there was great thoughts of success, and a lot of things didn't work. But I think the underlying the underlying theme here is that what eventually worked was to actually both build much better clocks, and that could actually measure time very precisely, and also understand planet precession, so they can basically combine the two things very accurately. And I think that every single time, the reason we, for instance, don't explode any more nuclear bomb in the States, because you can actually predict very, very precisely using a model exactly what will happen with a particular design without having to actually explode one. So I think that the paradigm changes completely. And by the way, I disagree a little bit with the fact that the paradigm have failed. I think actually many of the paradigms we have in the past have succeeded, albeit in a relatively, we've learned what are the confines of the paradigm and where they succeed. But I think that certainly the underlying model is that if you can build more predictive models, this is proven right in every single discipline, every single science over and over again. So I think hopefully we are on that path right now.
Well, I'd point out that the longitudinal problem was they knew beyond a shadow of a doubt that there was a large engineering component to that. They knew if they could develop a clock that was dead on accurate, that didn't shift with motion on ships, then they could measure longitude. I'm not quite so convinced that's the case here.
John, we're really running, it's been a wonderful conversation. We're going to have to draw to a close, so I'll get you the last question, and then Brenda, I'm sorry, we're going to have to do yours over maybe the next session or over the coffee break. So, John.
So thanks for the last question. Probably not quite as provocative as the last. John Wagner from Merck. So Bill, you brought up the concept of some of the key research findings not being reproducible with reproducibility rates as low as 10 and 30%. And with the idea of combinations, that's also factorial, right? So the what can go wrong is now multiplied as well. So the question for the panel is there some intermediate point between the science and the engineering of systematizing what's being done in basic discovery research that can sort of get us out of this problem of lack of reproducibility?
Well, I think part of the problem, and this gets a little bit off topic, but when I started out in the 90s, a paper was, I've discovered one thing and here are 6 lines of evidence that's true. So I'm trying to have multiple lines of corroborating evidence. Now in the quest to have what is referred to as the complete big picture paper that gets into the high profile journals, a paper now is 6 things, each proven one way. So it becomes sort of a house of cards. And so I really think that part of it is the sociology of how we put papers together and the kind of papers we reward. And I think it directly impacts on what you're referring to, which is the robustness. But the second thing is to do the kind of work that needs to be done to validate a target in preclinical models. A lot of that work really shouldn't be done by technique, PhD students, and postdocs. And so we probably do need to have another way of getting that work done, because in some cases I don't think it's completely appropriate for it to be done in an academic environment.
I just want to comment again on the reproducibility. We know some groups that share reagents, methods, they try and reproduce it as many of you do in other labs, and that adds to the robustness. I think in some academic labs, they don't even know what all the variables are, of course, and so it's very difficult to reproduce. It's not that it's an intentional cherry picking, but just sometimes just the rigor of doing the methods is just such a different mindset that it's very difficult to reproduce.
Just a final comment that it's also important to remember that these are diseases that are happening in human patients who have a wide variety of comorbidities, who have incredible differences in their own genome in terms of how they metabolize drugs and how they produce growth factors in the microenvironment that may support or not support the tumor. I think as Bill said, if you take a single AML and you take each individual cell and do the genomics of that, you'll find every possible combination of the mutations that you see when you do bulk sequencing of the overall tumor. And then you have the epigenetic changes, which may allow a particular mutation to be expressed or not expressed, et cetera. So I think these are wonderful, optimistic approaches. I think that also the challenges are going to be extraordinary as we continue to try and solve the problem. Anyway, it's been a really, I think, wonderful discussion. I'm going to ask people to come back at 10:10 so that we don't run too far behind schedule. Thank you.